Hello, everyone, and welcome to today's webinar titled, You Like Puzzles? Try to Put the Pieces in Cancer Together. This webinar is a part of the 7th annual event in the Cell Biology Virtual Event Series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Luminex, a DiaSorin company. For more information about Luminex, go to www.luminexcorp.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees in our live chat feature during the presentation. You can also find the live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits image located on the right of your screen and follow the process to obtain your credits. Now let's get started. I'd like to now present today's speaker, Dr. Christian Regenbrecht, co-founder and CEO of CELLphenomics. For a complete biography on our speaker, please see the Presenter tab located to the left of your screen. Dr. Regenbrecht, you may now begin your presentation.
Hello, good morning, good afternoon, and good evening, depending from where you're watching this broadcast. Thank you for the kind introduction, and of course, thank you for the invitation to Luminex that I can present what we have done in the past years in advancing patient-derived cell culture models and how we can put things together for better treatments and for the identification of novel biomarkers. So the title of my talk today is You Like Puzzles? Try to Put the Pieces in Cancer Together. This comes with reason. I think cancer is one of the most complex puzzles, speaking in terms of diseases. And it won't be a single technology, a single drug, or whatever that will work against all cancers. So you need to have a toolbox to get the pieces together, to match the pieces, and to turn data into information and information into something actionable. And that is what we hope to deliver, a piece of the puzzle that helps us understand the cancer problem. So when I say we, please allow me to introduce me and my two companies. So we have founded CELLphenomics in 2014. We are located at the Max Planck Center, the MDC campus in Berlin and Brandenburg, and have around 250 square meters of office and lab space. Currently, 20 people are working in the company, and our customers are mostly biotech and pharma. And during the years, we have an established biobank, a living biobank, with our complex three-dimensional models of about 450 models. So these are numbers from a month ago or so. So we have exclusive collaborations with hospitals in Germany, Austria, Switzerland, and the UK, where we get our tissues from. We work with our partners in national and international research projects like PRECODE or TUMOC, and we have a small study as well on sarcomas. And we expanded our business in 2019 with the second company, AAC Oncology. And in principle, AAC Oncology is working on the same set of technologies, yet the customers of AAC Oncology are solely oncologists and patients. So yes, we are delivering functional precision medicine to oncologists and their patients. And we keep that separated from the interests of the biopharma industry. So that's the quick introduction of the two companies. And our core competences is the establishment of high quality organoid models from cancer tissues. We support companies in drug development and biomarker identification. And we can aim for both target-relevant or disease-relevant models. Target-relevant models means we can look by genomic sequencing, but also by protein profiling for models that share certain properties. Or we can just go very old-fashioned by the site of the disease, like we can look for head and neck cancers, colorectal cancer, you name it. And with that, we can also provide guided therapy suggestions for patients and oncologists. So trying to rule out those treatments that will not work eventually and direct the treatment towards something more successful. Just a little glimpse about the timeline when this all has started. So in 2014, we founded the company as part of Oncotrack, which was a large IMI-funded project. And here we also started exploring the proteomic space together with Luminex and the TijiWest technology, which I will talk about later. And then we developed it. We got more partners. We got more understanding of what tumors do, what do tumors in regards of their treatment. We got awarded a patent in 2021, which I will be talking about today. And yeah, as you can see here, we are expanding our business in various countries. So the question that we need to answer is, what makes a good in vitro model in oncology? It comes with a certain list of must-haves and some nice-to-haves. Let's start with the must-haves. One of the most important things that you can imagine from a good model is that it is available, that you can obtain it. Then, and here I'm going again into the direction of the puzzle, the maintenance of key features of the donor tumor. So what are these key features? It's on the histology level. It can be immunohistochemistry markers. Then zooming into the cell, we look at the entirety of the DNA, so the genomic features. And then we zoom in even more to the key mutations, the SNPs that explain the addiction of pathways to certain genes. Then the model needs to be reproducible. So it must be made sure that if you repeatedly freeze and thaw the models, the data is robust and reproducible. So the last points belong very closely together because reproducibility can only be assured by certain robustness. Then let's look at the nice-to-haves. Most cancers are divided in molecular subgroups, and these subgroups need to be represented by the model, so representative for the cohorts. Then, of course, we want to learn from the models. We want to learn how a single gene, for example, a mutation, introduced mutation, plays down in the signaling cascade, so it must be prone to molecular manipulation. Then the compatibility with existing models like PDX models is a nice to have. And in our modern days, cost effectiveness, I think, is one of the top priorities for the industrial partners. So let's look at our models. The models recapitulate the donor tissue architecture. Most of you won't be pathologists, but I think it's easy even for lay people to see that here these sections of the donor tissue are very much alike with those of the matched PD3D model. This means the tissue architecture is basically obtained in our cell culture PD3D models. This similarity is so good that a pathologist was able, in a blind test, to match for 5 patients with breast cancer the right patient to the right model. And in sarcomas, rare cancer of connective tissues, in a blind test, 40 models were correctly subtyped. And by WHO definition, there are 120 subtypes of sarcomas. So that is a great thing, that these models rebuild the biology of the tissue. And here on the right side, you can see that basically also the morphology by the tumor site is very different and distinct. You have colon carcinoma looking different than these ductal shapes from a mama carcinoma. And you can see here this almost connective tissue-like cell culture from a sarcoma. So this is the space that we are using on the cellular level. And when I was talking about the robustness, we also see that when we cultivate our models, we did so with one for over 1.5 years continuously, and compared it with the donor tissue. The original donor tissue had 288 mutations, while after 1.5 years, the derived PD3D model had 293 mutations, meaning an additional 5 mutations, and a mouse model that was generated from the same donor tissue after 3 passages had 3,350 mutations due to the instability of the genome in the mouse. That, I think, is very convincing about the quality of models, and only if you have a good quality of models going in, you can collect valuable data. Together with colleagues from Eli and Lilly, we have developed a pipeline for semi-automated screening approach, where we seed out the organoids in a 12-well plate, make a single-cell suspension, put them by a liquid dispenser system into a 384-well plate, and put some fresh medium on it, allow them to recover from the procedure. Meanwhile, we are preparing compound plates with dilutions of the compounds that we will use. We will then stamp them over on the 384-well plates with the tumor, allow them to work for about 3 cell cycles, which is for most cases about 96 hours. And then we assay the plate with a cell-target glow assay, so ATP dependent consumption. The example workflow that we use is we obtain tumor and normal tissue, use the tumor for model generation, freeze and save the aliquot of the normal tissue, but also in best case a bit of the tumor. We have the donor consent, of course. We assign IDs for anonymization. We generate the models, expand them for at least 5 consecutive passages if we're doing it for biotech and pharma. We do it, of course, much earlier for patients because their time is more crucial. And usually when we do it for patients, it needs about 30 days from receiving the tumor tissue to delivering the results back to the patient and oncologist. So at passage 5 to 6, we collect the cells for QC, freeze them down, do that with our sequencing partner here in Germany, CEGAT. They confirm it's tumor, it's not. If the model fails, the experiments are ended. If yes, the models will be established. And so we can make sure that we deliver high quality material for high quality analyses for our partners or our own research. So how does the situation look today? The clinical guidelines for therapy of colorectal carcinoma. First of all, at the beginning, irrespective of any molecular cohorts, the progression of a tumor, especially in colorectal cancer, can take up to a couple of decades. Usually it starts with a small polyp that is residual in the tissue. Then the tumor advances stage 1 to stage 4, which is the full-blown metastatic cancer. And for the stages from a benign polyp to stage 3, it's cold hard steel. Surgery is the way to go. And only in the later stages, chemotherapy and in subsequent therapy lines, targeted therapies are used in addition to surgery. So you have the heterogeneity of cancer patients, and despite some minor differences, all are started being treated the same. So the problem is that with different molecular properties and molecular reasons for the tumor evolution, we cannot hope for one drug fits all approach. This is, I guess, quite clear. So what we need is the right drug for the right person at the right time. But therefore, we need to solve a puzzle. To solve the puzzle, who is the right person? Which is the right drug? Why will it work? Why can't it work? So this is where we jump in with our technology. We generate patient-specific 3D cell culture models. We call them PD3D. The literature calls them PDOs, some call them organoids. Then we combine the approaches of drug screens, omics technology, and gene editing tools to understand the individual cancer, what drives them, what is the Achilles heel of the individual cancer, and then come up with suggestions. That can be suggestions for cohorts of patients, but it can go down to the single individual patient that we are looking at. So, and if we go now onto the map with the organoids and zoom in there with the magnifier glasses, we can identify crucial points. We can mimic the location in the colon. We can mimic the advancement in time. We can go very, very, very specifically down there with the drug screens and identify groups and classes of drugs, but also within classes of drugs, single drugs that work in the individual better or not so good. Come up with the suggestions, and on the mechanistic level, which is what I think scientifically the most interesting part, we can manipulate those organoids and the DNA of those organoids to see what happens. So syngenic organoids to unravel the function of a single gene is something that we can easily do, and then analyze this, get this new genotype, understand the new phenotype, and find out what are the mechanistics behind it. So what I want to show you is we took a multi-regional sampling of a colorectal cancer patient tumor. Means we got a big bunch of tumor, and from 5 separated sites within the same tumor, we took material and grew organoids from it. We named them region R1 to R5, but here we just focus on region R1 and R4, which are, I think, scientifically the most interesting. Both regions share a set of mutations, KRAS mutation, PI3 kinase mutation, P53 mutations. But what we found out is that they were different in a single gene. There was an additional mutation in the SMAD4 gene in the region 1. And how did we find that out? Basically, by watching the cells, by treating the cells with drugs, and seeing in most drug classes, these cells behave very, very similar, but then in 1 or 2 specific classes, which I will come up with later, there was different response between those regions. Interestingly, the oncologists and pathologists didn't believe us at first. That was because they didn't find this SMAD4 mutation in the patient tissue. Well, what we did, we used ultra deep sequencing with a 2,000-fold coverage. And with that, we could also prove it as existent in the original donor tissue. So we were lucky, or we increased this population with the SMAD4 mutation, and that it was a mutation that in a clinical setting nobody was aware of. So that is just a little remark on the quality of sequencing in the clinical environment. So what does SMAD4 do? SMAD plays a role in TGF-beta BMP signaling pathway. You have here on the one hand the EGF receptor axis with RAS, RAF, MEK, and ERK. On the other hand, you have TGF-beta crosstalk with BMP, and the crosstalk player is SMAD4, which forms heterodimers with SMAD2/3 on the one hand and SMAD1/5/8 on the other hand, and then act as cofactors in the nucleus. So basically, that is the role. And what happens now with the loss of function mutation in SMAD4? An overrepresentation of the MAP kinase pathway. So let's look at the clinically used biomarkers for colorectal cancer today. And if we go along the EGF axis with RAS, RAF, MEK, and ERK, we found that biomarkers for response in colorectal cancer patients stage 4 is the RAS status, is RAS wild type or is RAS mutated. The BRAF status, again, wild type or mutated. Are we on the left side of the colon or are we on the right side of the colon? Both cases get a chemotherapy, and the location and the RAS mutation status decides about the addition, which could be plus cetuximab for RAS wild type or plus bevacizumab for the RAS mutant or right-sided colorectal cancers. But at the end of the day, this doesn't matter too much because there are too many non-responders. 40% of non-responders for the RAS wild type branch and about 25% of non-responders for the RAS mutation part. And we think this is not good numbers, but it shows very well and illustrates why today we still need systemic chemotherapy that kills all rapidly dividing cells just to reduce the complexity by killing as many cells as possible. But could that be any better in the future? We will see in a moment. So, guideline therapies. We were asking our questions, is SMAD4 mutation responsible for different drug response in colorectal cancer? And 2, would then SMAD4 be a suitable biomarker? Let's try to answer question number 1. I briefly introduced you into our method, and this is how we actually have done it for this tumor sample. We disaggregated the organoids, we used the automatic cell seeding, allowed the cells to reform into complex models for about 72 hours, and we added the compounds daily, because what you need to note is that these kind of inhibitors also in the clinic are replenished daily. So that is important to see and measure a really clinically relevant effect. And then after 96 hours of treatment, we did the cell titer glow readout. The factor that we are looking for is the Cmax value. Cmax is basically the maximum concentration that you can get in a human plasma sample, meaning the maximum amount of drug that can be transported to the tumor. And how can that come from the intake of the drug until it's solved in the bloodstream and delivered to the tumor? So let's see how these things connect to each other. We treated both region 1 and region 4 with a group of inhibitors, and we systematically went down the RAS signaling cascade. So we started with regorafenib, dabrafenib, termetinib, and ulixertinib. And you see there is no difference in regorafenib treatment between the two cell populations from the same tumor. With trametinib, the situation looks completely different. Trametinib is a MEK1/2 inhibitor. And with ERG, here you see the respective IC50 values. You see how close they are for regorafenib. And you see here we have a very good result and a not reached IC50, even when escalating beyond the Cmax value. So we decided if that was the only mutation, the SMAD4 mutations, the only difference between those cells, we used CRISPR to generate a CRISPR clone from the wild-type population, introducing the additional mutation that we have seen in R1. So we did the editing, we did the cell sorting, clonal expansion, confirmed it by sequencing, recultivating it, and then we came up with a group of CRISPR clones. We have the transfected and sorted R4 region cells, confirmed it, classical Sanger sequencing, and then we had 3 clones that we have interrogated, and the data that we have generated from all 3 clones was very similar and comparable with no significant differences. So the variant allele frequency was determined 100%, so it's homozygous. Here's just for the nerds with you, the sequences that we have used. You can stop the video at any moment when you want to look at those sequences a little bit longer, but I'm now going through this. And then we see that with not only trametinib, but also cobimetinib, which is another MEK inhibitor, and silumetinib, the results between region 4, wild type, the originally mutated region 1, and our clones with the inserted mutations are very distinctive and very different between R4 and R1. So, yes, indeed, the introduction of this additional mutation in the wild type clone, wild type region with 3 different clones changed the response for the sensitivity towards MEK inhibition. Question number 2, is SMAD4 a suitable biomarker? So what you see here in these waterfall plots, this is the list of the models that we have checked. And we have, as we have sequenced them, we knew which were wild type and which were mutated for SMAD4. And you can see here with the red bars, they are more in the upper side, except for silumetinib. By the way, that is also not a MEK inhibitor which has an approval for treating cancer, but for other indications. So that was also interesting. Same target, different way. So if we go through it, sorry, we have the sensitive on the left and the resistant models on the right side. And if we go now further down the road and do sample collections at different time points, and this is where the Luminex technology comes into play, we were able to isolate the proteins from our models and employ DigiWest. And DigiWest is a very interesting technology. What you do is basically you run a classical Western blot, cut out the lanes, cut out each of the bands, then you come up with like 96 stripes. You elute the protein that has been bound to the membrane, and then you couple it on Luminex magnetic beads, and as each of these bead sets has a different, let's say, identity, you can later pool them and put a PE-linked additional primary antibody on them. So then you can multiplex with the amount of protein usually used for single Western blot. You can now do, in theory, more than 800 Western blots. What we basically did is up to 200 Western blots in our models in one run. And this gives you very dense information, not only data, but information about the proteins and the post-translational modifications. And that is what I see as the big pro of this approach in regards to expression, because when you do RNA-seq, you only see the RNA and not is the protein there and is this protein activated, for example, by phosphorylation. So with this technology combined with what we have done, and we used proliferation markers, Aurora 1 and cyclin B, we were able to see that this plays out also in the BMP pathway. Again, SMAD4 is at the interface between those two pathways. So from there, we asked ourselves, can we become even more precise when looking at this biomarker? Because you saw that for selumetinib, we ended up with just like a 55% chance to catching it. And then we identified what we call the SFAP signature. SFAP is for SMAD4, FBOXW7, ARID1A, and BMPR2. So it's a group of 4 proteins that play together. And if any of these 4 proteins or the genes for these proteins is mutated as a loss of function mutation, we see the same result. So, as I said, models need to be representative. The frequency of SFAPs in our PDOs tested is ranging. Any of these mutated genes is in some accumulated 30%. If you look in the Cancer Genome Atlas, it's a little bit higher, 35% eventually. But I think you get it. Our 62 models mimic quite well the cohort of 594 models that are there in the Cancer Genome Atlas. If we now look at the same data and say, does it have a mutation in one of the SFAP genes versus a mutation in the SFAP genes? The positive prediction grows to up to 95%. And that is where we really would like to end up with our biomarker and the identification. So the currently clinically used biomarker, I explained it earlier, is the mutation status of BRAF. And for any of these 3 drugs tested, cobimetinib, trametinib, and selumetinib, we outperform with our new biomarker the predictive value of what is currently clinically used. And that is mostly owed to the DigiWest technology, which is based on the Luminex technology, as I have shown you here. Let's now dig a little bit more just into the analysis, because with this, we were able to see on the protein level how close the clones are together and how well they resemble region 4 in contrast to region 1. And with that, we were able to melt down the list of targets, of target genes, of proteins that could be useful for such a marker. And with that, we also were able to compare on a quite complex level the phosphoproteome signature between the original R4 region and the R1 region and their respective CRISPR clones. And there we could really, on the mechanistical level, see how this shifted away from an activation to a deactivation, and that higher Wnt signaling activity in the SMAD4 wild-type PDOs versus higher expression of anti-apoptotic markers in the mutated PDOs. And that, I think, is very astonishing, because usually it takes much more effort to get such a clear signal to noise ratio that you are able to identify a pathway that is clinically relevant, that can serve as a biomarker. And yeah, for such a relatively small company as we are, this was really a breakthrough with a method that also price-wise outcompetes most of the sequencing folks. So just to show you a little bit the details of not just these heat maps, but if we look here at the data we see in the MAP kinase pathway, there is no difference in MEK expression, MEK1 or 2, in ERK1, 2 expression. Where the differences is, where we fall down, is in the phosphorylation, something we would have never seen in sequencing. Same is true, there are no differences in the TGF-beta PMP signaling for SMAD1. It's the expression of the protein is there. It's just the activation. And the big difference you see, I guess, best in the mTOR pathway with PS6 and PMTOR, very different below the protein level or increased up to 300-fold measure in the region R4. So we could show that this was responsible for different drug response. And we were able to show that this is a suitable biomarker because it outperforms the RAS-BRAF status significantly. And therefore, our suggestion for the future of the treatment of colorectal cancer is using a technology that identifies the SFAB signature before giving the treatment, so you can spare the patient the chemotherapy and go directly into the targeted therapy approach. So in the outlook, we will generate PDOs syngenic for the other genes as well, and also test other cancer types for the SFAB signature. One of the most interesting tumor types there could be malignant melanoma. In parallel, we can perform retrospective clinical studies of those patients that suffered colorectal cancer and were treated with a MEK inhibitor. And of course, if we find suitable partners, we can do a prospective clinical trial on that. So in conclusion, MEK inhibitors should be considered as standard therapy for colorectal cancer patients where the tumor has the SFAB signature. You can read basically most of what I told you in an article published in July last year. And as I said, we have filed for an international patient for this marker, so we can always talk about how you can employ our marker in your patient cohort. I had the pleasure of presenting today to you, but of course this is team effort. So I wouldn't be here presenting without the great teams at CELLphenomics and AAC Oncology, but also with our partners from the NMI in Reutlingen, the Charité University Hospital, the folks at the Max Planck Institute, and the University Hospital in Rostock. So thank you very much. And I hope you have learned a little bit and I could entertain you a little bit. And there was some food for thought in regards of how we treat cancer patients in the future. Thank you very much.
Thank you, Dr. Regenbrecht, for your informative presentation. We will now start the live Q&A portion of the webinar. So if you have a question that you'd like to ask, please do so now. Just submit a question into the Ask a Question box. All right, let's get started. Our first question here is, would CELLphenomics 3D models and screening be helpful for patients that have been unlucky with previous cancer treatments that did not work?
Thank you for the question. And the answer is definitely yes, but, and the but is, we have specifically generated the second company, AAC Oncology, to serve the patient's needs. And we had many cases where we already successfully were able to direct treatment. mainly in rare cancers. This works very well, where guideline data is quite weak. Then it works very well in so-called CUP tumors, cancers of unknown primary, because if you don't know where the enemy hides, it's difficult to fight it. And with our method, we were able to not only direct the treatment, but help uncovering the cancer of unknown primary that later then become a breast cancer. So that is two of the main things. And of course, we have many patients asking us for help, and also oncologists where first and second line treatment failed. And people are basically on the last line that could save their lives. And here we work together with the oncologists to see and help and fuel some out-of-the-box thinking of drugs that have approval maybe for other tumor sites, and then we repurpose those drugs to find the right drug or combination of drug for each and every cancer patient. So yes, that is what we're doing, and this is what our job makes so interesting, and my entire team is so happy to be able to help people, because we can actually make a difference.
That's wonderful. Thank you so much, Dr. Regenbrecht. Another question we have here: could the 3D models and screening help with the drug discovery of both biologics and small molecule drugs?
Yes, we work with a list of, we've compiled a list of about 250 drugs that are approved and that we can test. Important is that we get some information of the range where we do the screening, and the expected maximum concentration in plasma, which is usually determined already in preclinical phases in animal models or in later phases in patient cohorts. And then we can help refining the drug approaches for also novel drugs. And currently, we are working on getting even better co-culture models, meaning immune oncology products can be tested, because we have mostly solved the problem of how to read out from patient-derived autologous blood cells and the tumor cells, so that we can really mimic also in vitro how immune oncology drugs work or do not work in a specific patient. So just a little spoiler, maybe from the next presentation I'm allowed to give on this channel, it's not all about PD-L1.
Great. Well, thank you so much. We have a few more questions here that we have time to answer, so we'll go ahead and proceed. This next question asks, any surprising insights you have found from looking at post-translational modifications in your 3D cell culture models?
Oh, yes, definitely. So, I mean, sequencing basically is a commodity these days, but there's so much more in that. I think I've also shown that in one of my last slides, that you do not actually see a change in the overall protein, and the regulatory mechanism behind response to MEK inhibitors or non-response to MEK inhibitors is just the phosphorylation, something that you cannot capture with the sequencing technology, and something that we are so happy that we can use DigiWest, because also other proteomic amines would consume so many cell material that it would take too long, and way longer than it does today, generating enough cells for the subsequent analysis. So with this targeted proteomics approach, it's a great trade-off. Of course, we don't find de novo things as we look into specific pathways, but when looking into those actionable pathways, which I think is the most interesting for patients, we are able to quickly understand. And if I say quickly, it's like when we prepare the protein to we get the results back, it's about 3 weeks. And quickly understand the biology that leads to treatment success or treatment failure.
Great. Well, it looks like we have time for one more question here. So this question asks, how can the CELLphenomics 3D model and screening data be adapted for other diseases such as autoimmune or chronic conditions?
Well, that is a really good question. Currently, we are a one-trick pony, but we are a pony that has perfected the trick. To be honest, there's so many things that we need to explore in the cancer space and refine in the cancer space, and so many ideas my team and I have to get better oncology models that we didn't spend too much thought on how to go into other indications, although we know that this would be worth looking into it. And whilst the company is growing, I hope that we can allocate more resources also to look beyond the cancer space.
Great. Well, that does conclude our time today. Dr. Regenbrecht, thank you for joining us. Do you have any final comments for our audience?
Yes, maybe. First of all, thank you again for listening and taking interest into what I had to say. But my comment is, if you look at the cancer puzzle, and no matter if you're into proteomics, into genomics, into cell culture, please accept that it won't be your or our technology or approach alone that will solve the puzzle. We have to work hand in hand for the sake of better treatment of these patients that currently, despite all progress in oncology research, don't have a chance. And that is something I hope for, that we join forces to get more functional precision medicine.
Wonderful. Well, I'd like to thank you again, Dr. Regenbrecht, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd like to thank our sponsor, Luminex, a DiaSorin company, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until September 20th, 2025. Labyrinths will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.
Goodbye. Thank you.